Hello, everyone. Welcome to today's live broadcast, Sex as a Biological Variable in Rodent Models. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots, the leading scientific social networking site and producer of educational web seminars. For more information, please visit www.labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of your questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Stephanie Murphy. Dr. Murphy received her VMD and PhD from the University of Pennsylvania. She then completed a postdoctoral fellowship in the Department of Comparative Medicine at Johns Hopkins and is a diplomat of the American College of Laboratory Animal Medicine. After completing her training, Dr. Murphy joined the Department of Anesthesiology and Critical Care Medicine at Johns Hopkins as an assistant professor. In 2003, she joined the research faculty at the Oregon Health and Science University, or OHSU, where she was engaged in research focused on cellular and molecular mechanisms underlying sex differences in stroke and directed an animal models core. While at OHSU, Dr. Murphy also served as the Associate Center Director of the Translational Modeling Program for the OHSU Research Center for Gender-Based Medicine and advised researchers regarding sex as a biological variable in experimental studies using animal models. Dr. Murphy is currently the Director of the Division of Comparative Medicine in the Office of Research Infrastructure Programs, a program office within the Division of Program Coordination, Planning, and Strategic Initiatives at NIH. I will now turn it over to Dr. Murphy for her presentation.
Thank you, Brenda, for that kind introduction. I'd like to thank the organizers of the 2016 Laboratory Animal Science Virtual Conference for inviting me to speak with you today about sex as a biological variable in experimental rodent models. For much of my research career, I have been actively considering sex as a biological variable in rodent models of ischemic and other types of brain injury. As a laboratory animal veterinarian, I've also had to address challenges, create opportunities, and provide research support so that other investigators could address sex as a biological variable in their experimental animal models. This next slide summarizes what I plan on reviewing with you today. I will start off by defining the terms sex and gender before discussing why one needs to pay attention to sex as a biological variable in biomedical research with rodent models. I will then describe some common myths and urban legends about sex as a biological variable and provide some examples of when sex matters in human diseases and experimental studies with rodents. Lastly, I will review some general reproductive biology principles, experimental strategies, investigative techniques, and available resources to be aware of when contemplating sex as a biological variable in rodent and other animal models. So before discussing why we need to focus on this issue, I need to start off by emphasizing that sex and gender are not the same thing, but are distinct terms that should not be used interchangeably. Sex is considered to be the biological result of the interplay between sex chromosomes and gonadal hormones. It is a biological quality that can be applied to both animals and to humans. Gender is a term that combines self and societal perception of a person's sex. It is a socio-cultural process that is generally applied to humans. However, there has been some recent discussions within the biomedical research community as to whether some aspects of gender could be applicable to animals, and if the definition of gender needs to be expanded to include animals. I've included here on this slide a table that further demonstrates this dichotomy between these two terms, where sex has genetic, molecular, cellular, and physiological aspects, while gender encompasses psychological, environmental, social, and cultural components. I also want to make a distinction here between sex-dependent and sex-specific effects. Sex-dependent effects include things like weight, height, basal metabolic rate, body fat, and muscle mass, things that are not as easily manipulated in our rodent models as such sex-specific effects that includes things like receptor responses, cyclical variation, neurotransmitter differences, cytochrome enzyme changes, and sex hormone-induced effects. These are elements that are a little more easy for us to manipulate in our animal models. Now, why do we need to consider sex as a biological variable? Historically, disease models have mainly used either single sex or have not clarified what the sex of the animals in the study have been. These practices were based on the assumption that disease mechanisms or treatment effects would be the same for both sexes. Some of the reasons why a single sex might be used is that one sex may have a greater sensitivity to the experimental manipulations that are being done in specific animal disease models. Commonly, investigators will propose using only male animals as a means of decreasing experimental variability caused by female hormone cycling. And I'm going to come back to this point later on in my talk when I talk about myths and urban legends. On the flip side, some investigators propose using only female animals because they want to avoid any inter-male aggression that can occur in some species and strains that make co-housing difficult. But in some species and strains, females may be as aggressive or even more aggressive than the males. Over the past decade or so, there has been increasing recognition that disease conditions, disease mechanisms, and therapy responses may differ between males and females. This area has been of considerable focus, and back in the beginning of the century, the Institute of Medicine actually focused on this topic and issued a report on the understanding of sex and gender differences and their effects on health and illness. And the overarching conclusion of this report is that sex and gender appears to matter from the womb to the tomb. However, reversing this single-sex approach in animal studies has been slower to change than in clinical studies, and there are a couple reasons for this. Unlike clinical studies, no legislative mandate exists requiring inclusion of both sexes in animal studies. There have been concerns about availability and cost of female versus male animals. Investigators need to have some basic understanding or knowledge of reproductive biology in order to think about sex as a biological variable in their studies. And lastly, limited research funding support and opportunities make it difficult to increase the number of animals in a study when considering sex as a biological variable. I'm going to come back to this last point again a little later in my talk when I talk about myths and urban legends. Funding organizations and journals are also starting to pay attention to this issue because it impacts on rigor, reproducibility, and translational relevance of research involving animal models. And you've heard some reference to this in some of the other presentations that have occurred throughout this conference. I list here, for example, that NIH has released a notice last summer in June about consideration of sex as a biological variable in NIH-funded research. I've listed here some of the key points from that notice, but the bottom line is that NIH expects that sex as a biological variable will be factored into research designs, analyses, and reporting in both vertebrate animal and human studies, and that a strong justification based on the scientific literature, preliminary data, or other relevant considerations will be provided in applications proposing to study only one sex. These updates have already gone into effect for applications being submitted to NIH for the January 25th deadline and moving forward with future applications. So what are some of the common perceived obstacles in considering sex as a biological variable in rodent models? Well, earlier in my introduction, I mentioned one common belief held within the biomedical research community that the estrus cycle renders female rodents to be intrinsically more variable than males. However, there really isn't any empirical basis for this belief. I want to refer you to a meta-analysis of 293 articles done by Prendergast and colleagues that monitored behavioral, morphological, physiological, and molecular traits of female and male mice. What they found was that randomly cycling female mice were no more variable than males on any particular trait, and that's shown here by this figure in the upper hand of the slide. What they also found was that the variability was substantially greater in males for several traits, as is shown in this lower figure here. Furthermore, they found that group housing actually increased trait variability by about 37% in both sexes. So the takeaway here is that estrus cycle may not need to be monitored when utilizing female animals. Again, it's going to depend on the research question that's being addressed. Another common perceived obstacle is the premise that cost and number of animals are automatically doubled when considering sex as a biological variable in rodent studies. However, not every experiment needs to incorporate sex, as when considering any experimental variable, you need to do a careful literature review first to see whether or not that variable is going to be important in your design and what others have seen respective to that variable in setting up these types of studies. It may be necessary, if there's not much in the literature to guide you, to conduct small pilot studies to directly examine whether or not sex might influence the experimental system being used and the outcomes you are evaluating. Not all time points or experimental groups need to be covered when thinking about sex as a biological variable in your experimental design. And you can use things such as factorial design to optimize your animal studies and reduce animal use when you're looking at sex and other variables in your experimental design. Now I'd like to give you some examples where sex and gender does matter when talking about human diseases. Women experience higher rates of adverse drug reactions than men do. Stroke incidence rates in men are greater than women at younger ages, but more women die of stroke each year. Nearly 80% of people with autoimmune diseases are women, and approximately 70 to 75% of patients with multiple sclerosis are women. But when men have the disease, they tend to have a worse prognosis than women. The incidence of melanoma is higher in women, but mortality is higher in men. And women have half the mortality of men in acute renal failure. And I could go on giving you more and more examples of where sex and gender are important, depending on the area of interest. This next figure shows the results of a recent analysis of journal articles looking at the proportion of studies using male and female animals. The blue bars represent studies using only males, the red bars those using only females, purple for those studies that use both sexes, and then gray for those that didn't indicate the sex of the animals used in their study. And there's really two striking findings here I want to emphasize about this figure. The first is that those studies that did report sex, male animals are predominantly being used in many of these research areas, with sub-exceptions being in reproductive biology and immunology. The second really striking finding here is that there are still research fields, such as general biology, immunology, and neuroscience, where more than 25% of the studies did not report the sex of the animal subjects. And in the case of general biology and immunology, more than 50%. And by not reporting the sex of these animals, it's really making it much more challenging for investigators to reproduce these studies based on what's reported. This next slide further underscores this female gap in the experimental studies involving animals. The purple bars represent the percentage of women in a population presenting with the various diseases listed at the bottom of the figure, whereas the green bars represent the percentage of studies that used female mice and rats in the studies. And what you can see is there is a disconnect between the purple bars and the green bars for many of these diseases. We are making assumptions and outcome predictions on these various diseases, but we're misrepresenting the populations that are being targeted. I'd now like to give you some examples from my own previous field of research, stroke, where biological sex has altered outcomes in experimental rodent models, and what has mirrored what has been observed clinically in the corresponding human disease. This slide depicts a commonly used rodent model of stroke, the middle cerebral artery occlusion, or MCO model. In this model, the middle cerebral artery, which is shown here, is occluded by passing a filament up through the internal carotid, the common carotid artery to the internal common carotid artery, the circle of Willis, until it reaches the point of origin of the middle cerebral artery. And in this way, we can cause either permanent or temporary blockage of this artery and study the outcomes. This next slide demonstrates how we evaluate infarct injury in this model. Brains are harvested and sliced into 2 millimeter thick coronal sections, and then they're placed in a solution of 2,3,5-triphenyl tetrazoline chloride, or TTC, and incubated. Viable brain areas will appear dark red as a result of reducing the TTC by mitochondrial enzymes, whereas injured areas will appear white. And in this model, we are interested in looking at the cortex and at the caudate putamen. Now that you have an understanding of our models and the measured outcomes, I would like to present to you some data from the stroke field looking at sex differences in experimental stroke. And the study that you see before you was done by the Al-Qayed lab. In this study, Dr. Al-Qayed subjected male, female, and ovariectomized female rats to 2 hours of middle cerebral occlusion, followed by a reperfusion period, and then measured infarct injury, the white areas that I showed you in the previous brain slices, to calculate a percent infarct volume in the two brain regions, the cerebral cortex and the caudate putamen. And what you see here, when you compare males, which are represented by the blue bars, to females represented by the green bars, that the female animals have a much decreased level of infarct volume compared to the males. However, when you compare the ovariectomized females to the intact males, their level of injury is comparable. And these studies would suggest that perhaps female sex steroids are going to be important to stroke outcomes. There are several well-known risk factors for stroke, 2 of which are diabetes and hypertension. And what this slide demonstrates that even in the presence of these 2 well-known risk factors that can create even greater injury in stroke outcomes, we still see differences in infarct volume between male and female animals. For example, here in the cortex, you can see that female diabetic animals have less infarct volume compared to the males, and the same is true for the hypertensive animals. Now, I've talked about sex as a biological variable at the whole animal level. This really needs to be considered as well when you're talking about the cellular level, and for many of our in vitro models, this is something to consider because individual cells have their own sex. And I show here an example from Dr. Minue Lu's work. She was interested in looking at the role of astrocytes in the response to stroke. And she used primary astrocyte cultures to look at various mechanisms and outcomes and the effects of different treatments. What's distinctive about her studies is that instead of harvesting cells from pooled collections of animals, neonatal rodents, she actually set up her cultures based on whether or not she harvested cells from male versus female neonatal rats. So she then exposed her male astrocyte cultures and her female astrocyte cultures to oxygen glucose deprivation, which is a way we can mimic stroke in the dish, and looked at cell death outcomes. And what she found was that female astrocytes had significantly less cell death than the male astrocytes. So we've been able to actually show similar findings in other cell types where we've set up the cell cultures based on sex. And this makes one think about some of the issues people have had in reproducing culture studies, because people used mixed sex cells, and it's not always clear what the ratio of male to female cells are. And you can see with the difference between the two, the different levels of male versus female cells in mixed cultures could lead to a lot of variability in the outcomes people would see in these types of models. Another point I'd like to emphasize with this next slide is this idea of sex stratification and how it could have impact on data interpretation. And I show here some work from Louise McCullough's lab, where she was interested in looking at how two different agents could affect outcomes in the middle cerebral artery model. And I'm not going to go into the specifics of the drugs and the mechanisms, but the point I want to make here is the difference in the responses between males and females. If you look at the vehicle-treated animals in this one example here, again, you will see that female animals have significantly less infarct volume compared to the male animals. What's really interesting is the response of each sex when they're given the drug that's being looked at in this study. In this case, the female animals actually have a worse outcome when treated with this agent compared to the male animals. And this is true in both examples. If Dr. McCullough had used both sexes but had pooled the data, she would not have appreciated a difference between her vehicle and her agent-treated animals for either one of these cases. And results like this might suggest why drugs that do well in preclinical studies, which have used predominantly young adult animals, fail in human clinical trials, where they start to see some of these adverse outcomes in one sex versus the other. It also limits us in finding potential treatments that might benefit one sex versus the other if we pool our data and we miss these types of observations. Now, at this point, I'd like to talk a little bit about reproductive biology and how it might be used to refine animal models for applications in research on sex differences in disease. I mentioned earlier in my talk that one of the reasons put forward for using males in some areas of studies was as a way to reduce variability that might be due to female hormone cycling. However, I want to point out some aspects of male reproductive physiology that could potentially be a source of experimental variability in studies in which outcomes would be relevant to both sexes. So I give here a couple of examples. Some male rodents are seasonal breeders and undergo significant physiological changes during the breeding season. For example, chinchillas have seasonal variation in testis volume. Siberian hamsters can have fluctuations seasonally relative to body weight as well as testicular size. And the South African spiny mouse can have fluctuations in testicular volume and mass, seminiferous tubule diameter, spermatogenesis, and plasma testosterone concentrations. I want to also emphasize that male sex steroids are not constant and can fluctuate on a daily basis. For example, testosterone levels are higher in the morning in young men. Mature male rats show a trimodal pattern over a 24-hour period. Mice may also demonstrate a diurnal rhythm of testosterone with a pulse release pattern. So the argument that females have hormonal variability, it's not just the females. Males can also show fluctuations in their hormone levels. Another point I want to make here is that mice, male sex steroids also decline throughout the middle years of life in many individuals. This state is increasingly recognized as andropause. Recent evidence emphasizes that the neuroendocrine aging in men becomes quite significant by the fourth decade of life. This is unlike the rapid and overt loss of estrogen in women. Loss of testosterone is gradual in men and can be clinically subtle. So again, when we talk about hormone fluctuations, it's not just limited to female animals, it's also relevant to male animals. Reproductive senescence is another important aspect to consider. This phenomenon occurs in both males and females and includes all aspects of age-related reproductive decline. Andropause and menopause are just one aspect of reproductive aging. I know many people use andropause, menopause, and reproductive senescence interchangeably, but really it's andropause and menopause are just one aspect of reproductive senescence. And this can vary quite widely between individuals, and it's referred to as the climacteric in human females. Now that we've talked about male reproductive cycles, I want to briefly review female reproductive cycles. This is something that may be more familiar to us versus the males. The estrous cycle is comprises recurring physiological changes that are induced by reproductive hormones in females. And the length and frequency of these estrous cycles will vary depending on the species. For example, mice and rats are considered to be polyestrous because they have repeated cycles throughout the year, whereas animals such as hamsters are seasonally polyestrous, where they may have repeated cycles during specific times of the years. The two major differences between the estrous cycle of rodents and the menstrual cycle of women come down to the endometrium and the sexual activity. Relative to endometrium, it is reabsorbed during the estrous cycle, but shed during the menstrual cycle if conception does not occur. And with sexual activity, females are sexually active during one phase of the estrous cycle, and this is referred to as estrus or heat, but can occur at any time during the menstrual cycle. This next slide summarizes the 4 stages of the estrous cycle and the changes that occur relative to the uterus, ovarian behavior. And I'm not going to go over this entire slide, but for example, when we look at estrus, one sees the greatest distension and thinning of the epithelium. We see maturation of the ovarian follicles, germ cell maturation and ovulation, and we see sexually receptive behavior. This is in contrast to diestrus, where we see the resorption of the endometrial lining. We see corporeal luteal growth activity and then regression at the end of the stage, but we don't see the sexual receptivity. This next slide just shows the pattern of hormonal levels that occur during the rat estrus cycle relative to estrogen and progesterone. And what you see here is that with estrogen, you see a peak in estrogen levels in mid to late proestrus. And this peak in estrogen precedes the peak of progesterone that we see in late proestrus. And then there's a smaller secondary peak of both hormones at the end of estrus, beginning of diestrus. Now, I'd like to spend the remainder of my time discussing experimental approaches and techniques that could be part of an investigator's toolbox when considering sex as a biological variable in rodent models, and can be also part of the toolbox for those of us that support research. I'm going to be talking about the availability of female versus male and reproductively senescent rodents, sexing of fetal and neonatal rodents, the issue of blinding studies relative to sex, staging and synchronizing female reproductive cycles, manipulation of sex hormones, some of the dietary considerations you need to think about in our animal models, as well as sex and anesthesia. So first, let's talk about the availability of female versus male rodents. Both males and females are available from commercial vendors. However, the cost may be variable between the sexes depending on the species and the strain and the age of the animals you want to look at. And I want to make a full disclaimer here. I did a very, very informal review of 4 major vendors, Charles River Laboratory, Harlan, Jax, Taconic. I went to their websites and I did a brief cost comparative analysis of age-matched rodents to see what the general trends and patterns were relative to cost. And what I found is that for immunodeficient mice, outbred mice and rats, and inbred mice and rats, female animals were either comparable in cost or a little more expensive than the males, again, depending on what age you were looking at. For hamsters and guinea pigs, male and females had relatively comparable costs across lifespan, whereas gerbils, the female animals, were either less expensive or comparable in cost to the males. And again, this was just a very informal look at what the cost comparisons were. Relative to the availability of reproductively senescent rodents, they are available through vendors, typically as retired breeders, or there may be species and strain-specific agent models that are available for purchase. More often than not, these animals are not sitting on the shelf, and if you're seriously interested in using reproductively senescent animals, you might need to make special arrangements or contracts to make sure that you have a regular supply of these animals. There are some reproductively senescent rodents available through resources supported by NIH. There are species and strain-specific aging models that are available through the National Institute of Aging, and the Office of Research Infrastructure Program also supports some resources that may make these animals available to you. Some academic institutions and research foundations may have their own private colonies. You may have collaborators and colleagues who may have aging animals that they might be willing to share with you, or you might consider setting up your own aging rodent groups. But if you do that, you need to make sure that you have sufficient space, time, and money, and that you plan for normal attrition. So for example, if you want 100 animals that are going to be 20 months of age, when you start your aging court, you may want to use twice that number in order to make sure that over the time interval that you're aging these animals, you will actually end up with the number that you need. Now, I mentioned before that in some studies people were able to set up sex cultures because they sexed neonatal rodents. We are able to sex fetal and neonatal rodents. We've been successful in sexing embryonic days 16 and 17 in rats and mice. And we use many of the same visual cues that we use in the adult animals. We look at genito-anal distance, and we look for the presence and absence of the testes and the uterine horn in these animals. And we do a lot of training with our technicians to make sure that they're comfortable sexing fetal and neonatal rodents. Like many skills, it just takes practice. While we're training people to learn how to sex fetal and neonatal rodents, we validate their training by actually doing PCR to confirm that they've correctly identified the sex of the animals. And we do this by either looking for the presence or absence of the SRY gene, which will be in the male animals, and then we have a background gene that would be present in both sexes. And you can see here, it's very clear, the pattern between the male and female animals. And when we use this PCR technique to validate the training of our technicians, we find that our staff are about 98% or better at identifying the appropriate sex in these animals. This type of backup PCR can also be of use when you're doing cultures if you'd like to double check and make sure that the cultures that you're using are the sex that you think they are. This can also be helpful if you have to combine genotyping and doing things like single culture. You can also confirm the sex of the animal. Now, a common question that comes up when I talk about sex as a biological variable is how do you blind for sex? And again, in several of the presentations throughout the conference, you've heard how important it is to randomize and to blind in your studies. And in order to blind for sex, you really have to take a team approach. And I give here an example from my own research program where we planned out different parts of the study so that we could blind the eventual evaluation by sex. We started out with a technician who would assign ID numbers to the animals and then randomize the animals to the various treatment groups. The next person in the chain would then take these animals, not knowing what they had been treated with or how they had been manipulated, and doing additional experiments, in our case, the middle cerebral artery occlusion model. They would collect the physiological data and the samples, and then those samples and physiological data would be handed off to the next person who would be blinded to both sex and to treatment and would do, say, for our example in our case, the infarct volume analysis, or would measure various analytes in the blood, or look at different compounds in the tissue. But they, again, would be blinded to the experimental manipulations and the sex. Now, for a lot of what we do in our lab, we would measure sex hormone levels, either to validate manipulations we've done or to see how hormone levels correlate to our outcomes. And since we have a limited blood volume in rodents, a lot of times people could kind of guess what sex we were measuring the hormone levels for. So we oftentimes could only blind for experimental treatments. But the point is, we did as much as we could to blind as far as we could in the process relative to our outcomes. Many times the data would be analyzed and then handed back to a third person who would actually break out the data relative to the code for the different experimental groups. So again, you can be creative in how you approach blinding for sex depending on your model and the questions that you're asking. Some other skills or resources you might want to keep in the back of your mind are ways of staging the female reproductive cycles. This could be useful in establishing timed pregnancies for sex-specific neonatal and primary culture models. It may also reduce experimental variability if you're using similarly staged females, again for those few traits where variability due to hormonal cycling might be an issue. How you stage will be dependent on the species and the strain. You can discern the different reproductive cycles either using visual and behavioral signs, vaginal impedance, vaginal cytology, steroid levels in the urine or the serum, or reproductive tract histology. And I'm going to talk a little bit more about some of these techniques. Vaginal impedance is commonly used, but there are some limitations. It will only often detect a specific reproductive cycle stage. It may not tell you all the different stages. There is some conflicting data in species, rat and guinea pig, as to when peak impedance occurs. Vaginal cytology is considered to be the gold standard in terms of staging animals relative to the different reproductive cycle stages. This can be done either by collecting cells in a wash or by swabbing. Each reproductive cycle stage is characterized by specific types and quantities of cells. But I should caution you, the length of the reproductive cycle stage will vary among rodents, and that will impact how frequently you may have to sample in order to narrow down the stage of interest that you want to work with. This next slide just shows some representative vaginal cytology smears, just to give you an idea of what the differences are between the different stages. For proestrus, it's characterized by these small, round, nucleated epithelial cells that predominate. For estrus, it's characterized predominantly by these irregularly shaped cornified squamous cells. For metaestrus, you predominantly see these leukocytes and a few cornified cells. And then for diestrus, you see predominantly leukocytes and a few of these round cells as you transition to proestrus. Synchronizing female reproductive cycles is another potential skill you could apply to your research design. Why you would synchronize? Basically for the same reasons why you would potentially stage the female reproductive cycles. Again, it's useful in establishing timed pregnancies for sex-specific neonatal and primary culture models, and it may reduce experimental variability if using a synchronized female. This can be done naturally. You can take advantage of the fact that estrus synchrony can develop in group-housed female rats after 3 to 4 ovarian cycles. This is comparable to the dormitory effect that's been described in women. We can also take advantage of certain pheromone effects, for example, the Witten effect in mice, which results from the exposure of female mice to a male or male urine, and this induces estrus in group-housed females. We can also, again, depending on the species and the strain, do this manipulation artificially by using either female sex steroids, gonadotropin-releasing hormones, and their agonists, gonadotropins, prostaglandins, and radiolytic agents. Now, depending on your question, you may actually want to control hormone levels. You may be asking the question, are sex steroids going to be important, or are there innate differences, or they don't play a role? Ovaries are the main source of female sex steroids, estrogen and progesterone, whereas testes are the main source of male sex steroids, or androgens. I should point out that the adrenal glands can also produce some sex steroids, but predominantly the gonads are a large source of these sex steroids. In order to significantly lower endogenous sex steroids level, you may have to do either a gonectomy or a combination of gonectomy and adrenalectomy. And I should point out that ovariectomy and adrenalectomy are considered to be major surgeries because you're either penetrating a body cavity in both cases, or you're producing substantial physiological impairment in the case of adrenalectomy. With orchidectomy, this can be either a minor or a major surgery depending on your surgical approach, whether you're doing an abdominal approach or a scrotal approach. However, you have to justify doing more than one major surgery if you're combining gonadectomies with other major surgical manipulations in your experimental design. So that's something to keep in mind as you move forward with setting up your experiments. If you want to look at hormone levels or you want to control hormone levels, not only can you reduce the levels, but you can selectively add hormones back if you want to see if it's estrogen versus progesterone that might be important, or testosterone versus DHT. And you're going to have to be skilled in different ways of administering these hormones. These can include parenteral injections, sub-Q, IP, IV, or IM injections. You can also use subcutaneous or IP implants involving either osmotic pumps, which I show here, silastic implants, which are homemade pumps, or commercial implants. Your choice of vehicle is going to be very important if you're going to be administering hormones to animals. Hormones are fat-soluble, so if you want to give an intravenous injection of hormones, you're going to have to look at a vehicle like cyclodextrin, which will allow you to do that. Vegetable oils will allow you to dissolve the hormones and to be able to give sub-Q and IP injections. But if you're going to be measuring hormone levels, your choice of vehicle is going to be very important. We found out the hard way when we switched from sesame oil to corn oil that that actually caused confounders for the hormone assays we were using. And we were getting all sorts of strange readings and variables in our assay. So when we switched back to the sesame oil, we actually were better able to measure our hormone levels. You can also use biodegradable matrices, which is used in some of these commercial implants. Again, if you're thinking about sex as a biological variable, you may want to measure hormones. This can be done in your lab. There are numerous kits available. Many institutions have core or core services available that will do the measurements for you for a fee. You may have colleagues or collaborators who could do the measurements for you. And there are vendors or commercial laboratories who will do it for you, send out the samples, and then they return the results to you. You can look at hormone levels both in the blood or in the urine. There are radioimmunoassay kits available, HPLC methods and TLC methods you can use. You can also measure hormones in the tissue using HPLC methods. Now I want to get to this issue of dietary considerations, and this has more to do with estrogen and estrogen-sensitive models. We know that estrogen administration can alter histological and functional outcomes in experimental rodent models like stroke, TBI, Alzheimer's disease, and EAE. Phytoestrogens, which can be in the diet, exhibit both estrogenic and anti-estrogenic effects and are found in most soy foods. And many of our commercial lab animal diets commonly use soy meal as a protein source. There have been studies reported that have shown effects of dietary phytoestrogens on several tissues and organ systems. And we know that dietary phytoestrogens could potentially modulate physiological and behavioral responses in estrogen-sensitive rodent models. And I just, again, want to emphasize this point. I show here 3 different rodent diets, and the total isoflavone level is shown here. And you can see that this diet has 2 to 3 times the isoflavone levels as this diet, and this diet has about twice. So if you change diets, or if you're not paying attention to diet, you could be blurring out some of your results or decreasing the sensitivity of what you're looking at. The last topic I want to look at and discuss with you is anesthesia. Male and females respond differently to anesthetics, and the differences in anesthetic response could be either sex-dependent or sex-specific, depending on the mechanism. So anesthetic choice in rodent models looking at sex effects may be a potential confounder. And I give here an example. In diffused traumatic brain injury study, female rats were shown to do better than male rats, but the outcome was very dependent on the type of anesthesia. The degree of the difference they saw between the sexes dictated what the background anesthetic was. So what they found was that isoflurane was protective in females, so they saw the greatest difference between the sexes. But pentobarbital was deleterious, and so they saw less of a difference between males and females. Halothane seemed to have the least impact on sex-related outcome in this study. So you may need to think about making adjustments in your anesthetic protocols for sex, again, depending on your research question. And I give here another example, pentobarbital. Male mice are more sensitive to the anesthetic effects of this drug than females. So when rats were given the same initial anesthetic dose, they found that males required more supplementation to maintain clinically comparative anesthetic deaths compared to the females. So again, just another factor to think about. Now, in the last few minutes of my talk, I just want to share with you a couple of resources where you can get additional tools, techniques, and information that can help you in thinking about sex as a biological variable when designing your studies, or if you're providing support for studies where this is a factor. And I point here to the NIH Office of Research on Women's Health. I list the website here. They have some workshops that have been video conferenced and are available as webinars for you. References, they do have some training materials and other resources that may be useful to you. Another great website that I frequently go to is the Gendered Innovations website. They provide definitions for different terms that are used in the field. They also provide some methodology, checklists that may be helpful to you in thinking about how you're setting up your experiments and how you're interpreting your data. The other thing that I like about this website is it presents different case studies in different fields of science and thinking about these questions and where it may be important, and again, in how you're interpreting your data. And then one last website I'd like to point you to is the Organization for the Study of Sex Differences website. They have an annual meeting, and oftentimes we'll put some of the presentations from that meeting there, as well as some other resources and training materials that might help you in thinking about this question. And then lastly, I want to share with you a sex-gender toolbox for experimental scientists that I often refer to, and I think really helps us to think about this variable as we would any other variable. You want to develop your knowledge of sex and gender issues. Do a careful literature review, just as you would for any experimental variable that you're thinking about in your design. Are there known sex differences or gender disparities for the phenomenon you're going to be studying? Avoid using the terms sex and gender interchangeably in your writing. That way we will create less confusion as to what it is we're talking about. Discuss sex and gender where appropriate. Always report the sex of the cells, tissues, animals, or subjects you're using. And I know this has been a common message in some of the other presentations that you've participated in during this conference. If you're using one sex, justify why, explain why, and note the limitations in your discussion. And always discuss the possible sex-gender implications of your findings. You might want to introduce a small intervention. Do a small pilot studies to examine whether or not some elements of sex and gender may be important in your model system. Add a hormone to one of your cultures. Include male and females in your key experimental groups. If you're working with both sexes, consider stratifying your data just to see if anything interesting comes out of that. Report what you find. If you don't see a sex difference, that's just as important to share as if you do. We want to raise the profile of sex and gender issues, and we look to each other to be advocates for this. As a reviewer, you can ensure that applicants and authors identify and justify the sex of the materials used, and make sure that the terms sex and gender are used appropriately. Ask questions of colleagues and trainees. Have they considered whether or not sex or gender might be relevant to their work? And in closing, I just want to emphasize again, sex and gender matters in many clinical diseases and rodent models. And again, I want to quote the Institute of Medicine report. It's important and it matters from the womb to the tomb. Sex and gender effects are understudied and not well understood. This could open up some new research directions and innovative funding opportunities for several of us. That's how I initially got into this field, is we saw something unusual and we investigated it and we realized it was a sex-specific phenomenon. Also, looking at sex and gender effects goes to the question of reproducibility and translational relevance. If you don't know the sex of the animal, how are you going to reproduce the study and the findings? If you don't know what the male to female ratio is in a pooled analysis, again, how are you going to replicate those findings? And how are you going to continue on to apply it to clinical patients? And then lastly, I want to emphasize that there are experimental strategies and resources available to all of you for considering sex as a biological variable in rodent models. So with that, I'm happy to take questions now. And if we don't have time for all the questions, I have my email address here in case people want to follow up with me with additional questions. Thank you.
Thank you, Dr. Murphy, for bringing us that informative presentation. Before we get started on the question and answer period, I would like to remind our audience and attendees how they can submit their questions. So questions can be submitted by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can. I just want to give our audience a few minutes to type in their questions and see what they have to ask you. It was really very relevant information that we have here today, and I'm sure we'll be hearing some questions from our audience. I do see a question. Dr. Murphy, an attendee asks, over the term of your career, since you've been at this for so long, have you seen a shift in more experiments and researchers looking at gender, whereas they didn't before?
Thank you for that question. I've been engaged in looking at sex as a biological variable now for close to 2 decades. And I would have to say really in the last 5 to 7 years, I've seen more of a shift. I used to be at study section. I would bring this question up all the time, and people would roll their eyes like, oh, there she goes again. She's talking about, are there male animals, are there female animals? But I'm finding more people are starting to ask those questions as well. In some journals, I'm seeing them require better reporting. The ARRIVE guidelines actually has made a big impact in making sure that people report the sex of the animals used in their studies.
Great. We have another question. An attendee asks, do you think genetic rodent models, such as for Huntington's disease, should be looked at in a gender-specific manner?
That's a great question. I'm going to have to speak in more broader conceptual terms because I don't have expertise with Huntington's disease. But I think it's always useful to ask ourselves, how does this disease present in both sexes? What are their responses? And one thing I caution to a lot of investigators is that even if you do a pilot study to see if there is a difference between males and females, and you don't see one, there could still be differences in the pathways each sex uses in responding to an insult or in developing a disease that could lead to the same outcome. But because they've used different pathways, if you're trying to target potential pathways for interventions, you need to be aware of those differences. And I also want to make a distinction here that considering sex as a biological variable is not the same thing as studying sex differences. You can consider sex as a biological variable, and based on that consideration, you may choose to focus on one sex or to include both sexes. But again, sex differences research is not the same thing. It's going to be driven by the questions that you ask, the hypotheses that you ask in terms of your experimental design. Now, I'm not, again, familiar with Huntington's disease, so I don't know the literature. I would do a literature search to see if anybody has even thought about whether or not there are differences in men versus women. If there's nothing in the literature, that could potentially be a flag to say, hey, well, wait a minute, let's just make sure that if I'm doing all this work in male animals and the outcomes and the assumptions that I'm making will be also relevant to female animals and eventually to female patients.
Great, thank you. We do have another question. Do you have suggestions for using sex or gender in neurological disorders?
I'm not sure I understand the question. That's a very broad question. There are some review articles available, and they're listed at the Office of Research for Women's Health website that talks about tools and strategies when thinking about sex as a biological variable in neurological diseases. I'm happy to email that reference, or if you go to that website, you can find that reference that provides some tools and resources in thinking about neurological diseases relative to sex and gender.
Great, thank you very much. I don't see any other questions coming in from our audience right now. We're getting close to the end of our time. I'd like to turn it back to Dr. Murphy for any final comments or thoughts that she has for our audience today.
Well, again, my favorite conclusion is, again, the quote from the Institute of Medicine report: sex and gender matter from the womb and the tomb. If you think about it, what is the first question we ask when a child is born? Is it a boy or a girl? And yet we don't really ask those questions of our experimental studies. Did you use male or female animals? So I encourage people just to put that on their radar and to think about sex as they would any other variable when thinking about their experimental designs.
Great, good information. Thanks so much for bringing that to us. I want to remind our audience before we go that this presentation is approved for continuing education credits. Please click on the free CE credit button in the bottom left corner to obtain your credits. I want to thank Dr. Murphy again and labroots.com for making this web seminar possible. Today's webcast will be available for on-demand viewing through August of 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay, and we certainly invite you to forward that email to colleagues who may have missed today's live event. Thank you very much for joining us. See you next time. Goodbye.